Good. So thanks for the introduction. Uh, if you were not looking forward to hearing about metrics uh, and how to calculate cyanobacteria populations, then I would say you're in luck because I'm not going to be talking about that. The talk that I'd planned to give did not make it onto the latest agenda. So I will start off with a different title. And this is some lessons learned about healthy lakes. And the reason I want to talk about this is we've learned a lot about cyanobacteria from looking at lakes where they're not considered to be a huge problem sometimes. And also many of the techniques that we're now using in our monitoring of cyanobacteria come from some of the things we've learned. So I thought I'd share those with you. Sure. First of all, some of this goes back quite some time when we first wondered whether we had microcystins in our lakes in New Hampshire. So we set out to do a synoptic study of the lakes, looking all over the state at different types of lakes. We called it a 50 lake study. It actually involved more than 50 lakes eventually. But basically what we found is, if you see the plot here of lakes and the amount of whole lake water microcystins, every lake that we looked at, even those lakes that had more than 12 meters secudisc, ultraligotrophic, up to the most eutrophic that we could find. And we sampled them in the middle of the lake. So we weren't looking for the worst conditions. We just sampled what we thought would be a good sampling point. But all of those lakes were positive. The reason they were positive is the lab I was working in was run by a biochemist at the time. And when we said we couldn't detect the toxins, he simply said, well, we have a technique in biochemistry we use to correct for that. And we speed vac, which is a type of freeze drying of your sample to concentrate it. So once we did that, we had no trouble detecting the toxins. And so the lesson that we learned from that first study was basically that toxic cyanobacteria were present in all of our lakes in all trophic conditions that we looked at. And the average lake in this system, I should point out, is probably more oligotrophic than even mesotrophic. So it's low trophic condition lakes in New Hampshire. One of my colleagues and friends, Rob Peters, wrote a book about the ecological implications of body size. What I was impressed with in this book, he compared the body size of bacteria to the body size of elephants and whales, and found that they had many things in common. And it was just a sliding scale of body size predicting a lot of things, everything from reproduction to respiration. And what I learned from that was that in ecology, you can learn a lot sometimes from a very simple metric such as size. So when we did our sampling in those lakes, one of the things we did is divide the lake water into whole lake water and then pour it through a 50 micron mesh. And the reason we did that is we knew that 50 microns was roughly the upper size limit for the filter feeding zooplankton, so that we'd be at least able to see some ecological relevance to the results. When we did that, one of the big surprises is that, and this red line shows you the 80% the 80% level. And basically what we found is in over 90% of the lakes that we looked at, more than 80% of the microcystins were in the less than 50. And then on the right-hand side shows you of that less than 50, most of those were less than 2 microns. So they were in the pico cyanobacteria size. So that we learned something from that the small cyanobacteria are not only important, but they can actually be more important for some things such as microcystins in the lakes we were looking at. The other thing is that, and this confirms what people in other countries have found, such as in Italy, The Cyanobacteria actually tend to be more abundant, relatively abundant, in the low trophic level lakes. So they're present in all of the lakes, but they make up a larger proportion of the toxicity in the small lakes. I mean, in the clear lakes. So the lesson overall then from that was that if we want to understand toxic cyanobacteria in our lakes, we have to really consider all the cyanobacteria and not just the obvious cyanobacteria. So we divide them into BFCs, or bloom-forming cyanobacteria, which generally are larger than 50 microns, and the PCYs, which are the picocyanobacteria, which tend to be 2 microns or smaller. Now, this division, this arbitrary size division, wasn't really created by us. When you think about it, it was created by the system. That is, the zooplankton in the system then sort of divided the world up into those edibles and inedibles. So when we look at it that way, we think then the lesson is, if cyanobacteria toxins are going to be considered, we need at least 2 compartments. One is the BFCs, which we know are highly toxic, but not very edible. And the PCYs, which are small, have some toxicity, but are edible, so that they're probably moving through the system at a faster rate. So when we construct then our model of how the lake looks, first of all, we have the picocyanobacteria, or the edibles. And this is from the perspective of cyanotoxins. And these edibles then move into the grazers, which then are moving up the food web, depending upon the composition, and ultimately end up somewhere near the top of the food web, in this case in loons that are eating fish. And this is sort of a classic food chain. So the small edibles, which both in terms of the toxicity, but also in terms of the signal that you get from the fluorometry, are actually then really important for the food web. The second is that the large forms, the BFCs, or the bloom-forming cyanobacteria, are largely inedible, except at times under stress. What we see is things like this occurring with Vornicinia, is that they can begin releasing their cells from the colonies. And when they do that, what we see then is there are some coarse feeders, such as Cereodaphnia here and Diaptomus. And those coarse feeders then can also feed into the food web. So some of this may get into the food web. And this then is, oops, I'm sorry. Let me, some of this then is actually going to then end up in what we call the minor side chain of the food web. And then the other, of course, is that the BFCs, true to their name, create surface blooms. And these surface blooms are often observed by scientists, but also by anyone using the lake, as you can see here. So this is what we most often see. So my message then is that we appreciate blooms. They're very important. They're often highly toxic. They're often responsible for reported acute toxicities that we see of pets and wildlife. But they also capture the attention of the public and the scientists. And we just want to remind ourselves that we already know then that lakes are complex ecosystems, and that blooms are only a part of that system. So it's just a reminder that although this is very important, there's more going on than just that. So just to back up a little bit as to where we went from what we learned so far, is in 2007, and then later we published some of this in 2009, there was a report of a high incidence of ALS, amyotrophic lateral sclerosis, around a lake in New Hampshire called Lake Mascoma. It turned out not to be the only lake in which this occurred, but the ALS clinic at Dartmouth Hitchcock Hospital was keeping track of where people lived. And they simply said, isn't it curious that near this lake we have about 25 to 28 times the incidence that we would expect normally for that region. And they were clustered around this particular lake. So we said, well, that's a correlation, and it doesn't really prove that there's a causal effect of that lake on the ALS condition. So then that was followed in 2015 with a study of an epidemiological study of liver disease, non-alcoholic liver disease, and water quality in lakes. And a positive correlation was found between the likelihood of having blooms in lakes and liver disease. Again, just a correlation. And similarly, in 2016, 2017, Nate Torbick and some other folks, I was involved in this as well, looked at the Northeast lakes and found that ALS clusters also seem to be statistically linked to the cyanobacteria in lakes. Again, not necessarily a causal relationship. So one of the interesting things that came out of this is that if there was more than a statistical link, then the influence of these lakes was extending out in Torbick's study as far as 10 to 30 kilometers. So that suggested an interesting radius over which then somehow the water quality had to be affecting people. So of course, we weren't the only ones to think of this. Was one of the likely ways this could happen, if it were to happen, would be through airborne toxins. So that started us actually thinking about aerosols. And most of the aerosol work has been done in marine systems where, in fact, aerosols are generally created by wind events. And so we thought, well, wind is less likely to be an important factor on lakes, so maybe there's less aerosols. Sometimes these are visible. This is shown here for a lake outlet near in the town that I live in. And this is, sometimes you can see them at night, the aerosols, and sometimes you don't see them. So the aerosols differ in the amount of water that they hold and the particles that they have, and the chemical constituency is quite variable. So we already know that aerosols are capable of transmitting bacteria and viruses and waterborne diseases such as Legionnaire's disease. We also realize that it's a potential sort of long distance link between systems, whether they're lakes, oceans, or the terrestrial system. So this set us off on a sort of a quest to learn more about potentially aerosols coming out of lakes. And Amanda Murby McQuaid at Murby at that time was a graduate student in my lab working on her PhD. And she and I spent some time developing a system to collect aerosols. And we're interested in developing a low-cost system, because we didn't have money primarily, a low-cost system to collect the aerosols in the way that other studies had collected, usually on a filter, not only, but usually on a filter, and then pump air from the water area up through this filter, and then let this run for a finite amount of time, such as 4 to 8 hours, collect the filter and examine it under the microscope for cells, and then measure it, extract the toxins from it. And that's what we did. We also said, it would be helpful to also do this under more controlled conditions in the lab. So we set up bottles in the lab and put the same lake water in those bottles and ran them. And we compared the results of the lab and the field. And the lab was a little bit higher, but overall there was a good relationship between the two counts. So what we learned from that was that there were aerosols coming out of these lakes, and that most of the aerosols we were looking at were in the form of Cyanobacteria cells. Okay. So if we go back to our original model we looked at, now we have to add in another portion, which is that the picos not only go through the food web, but they can also bypass this, entering into the air and then perhaps affecting wildlife, or if the air went in another direction, it could be affecting people, or it could be transported some distance, we don't know. So that's what I refer to as the aerosol bypass in terms of how we look at the toxin transport. Kate Langley was a master's student in my lab until last 2 years ago now. And she finished her degree working on using this aerosol collector, and having refined it somewhat, she also made it more compact so that we could run replicates more easily. It's called the CLAM, Compact Lake Aerosol Monitor now. And it also used the same basic technique of having the air pass through a filter, and then the filter was analyzed for toxins in cells. What she learned from that is that in the 8 to 9 lakes that she looked at, she did both day and night studies in all of the lakes. She found that they were toxic, both the microcystins as well as the cells were being emitted both day and night. And in some cases, like the cells, were actually generally higher numbers at night, and that they weren't really dependent upon the wind. So that we have aerosolization going on, but it's not a wind event. Secondly, that these toxic aerosols occurred in lakes. Generally, these lakes were mesotrophic to oligotrophic, so they were low trophic conditions. And what she found was essentially a confirmation of what we'd found earlier with the picoplankton. That is, that the highest percentage of the toxins in the lake were actually aerosolized in the more oligotrophic lakes. If you remember, the more oligotrophic lakes tend to have more of the picoplankton, so it made perfect sense, although no one had measured that.
3 minutes left.
What came out of her work, thank you very much. What came out of her work was the first model to explain how this works. What she found is, in fact, running a 2-parameter model, she was able to show that temperature differential, the difference between the air and water temperature, accounted for about half of the variation that we saw. But if you add water toxicity, you can explain 90 to 98% of the aerosolization of the cells. So this was a pretty successful model, and it's one we want to follow up on. But what about the mechanisms? And so we set out to determine whether we also have more dissolved than we've been measuring. So we set up in the lab and measured with purified microcystins and purified BMAA in water, and measured then what happens if you run them through water traps. And so we came up with a new CLAM that measures both particulate and also dissolved toxins. And this is the results from the Charles River project we did. And this showed, interestingly enough, that the dissolved microcystins were about 2 to 4 times that of the particulate microcystins collected in the air. And that this then, for BMAA, was even higher, that 4.5% or 4.5 times as much in the dissolved form as in the particulate. So these traps then are important in learning more about the aerosols. We have a study that, in fact, Anne Ewart did for her master's. She's defending her thesis actually tomorrow. And she determined that, in fact, the lettuce and radishes were actually getting more of the toxins from the aerosols coming from the lake than they were from the water that was used in this hydroponic system. So the last thing I want to show you is now we're beginning to think about aerosols in terms of emissions. So if you look at the lower mill pond, this should say, which we worked at on the Cape, and you think about it, this happened last night, actually. I made some rough calculations. So these are truly back of the envelope calculations. And basically what I did is try to determine, based on the average amount of microcystins coming out of the lake, how much was that represent if you corrected it for the volume of the lake and the surface area of the lake? And basically, I came up with a value which I say is very, very rough, but it's about almost 30 grams of microcystins per day that one lake is putting into the air. So another way of looking at these aerosols is actually think of them in terms of air pollution as well. And then the question is, where does that go? And basically, I want to end with this, which is that we need a lot of more research before we can get to human risk evaluations of this. It's more of a very important scientific finding, but to get there, we need to do more. We need to look at models. We can predict the aerosols, and both what I've shown in this picture is the smoke represents then the dissolved, and the other is the cells coming out. We need to look at multiple toxins. We need to determine how long they last once they're in the air. And then we need to be able to model where they move to, just as we've done for things like mercury and other pollutants. And then we need to eventually get to the point of estimating the health risk for humans and wildlife. And I'm trying to end this, and I will. There we go. Thank you very much. And a lot of people involved in all this work, and I thank them all.